you so much for having me here today. Our first speaker of the day, and it's my pleasure to introduce Joana Miranda, who is the group leader of the Advanced Cell Models for Predictive Toxicology and Cell-Based Therapies group at the University of Lisbon, and she's also Associate Professor with habilitation of the School of Pharmacy at the University of Lisbon. She obtained her PhD in biotechnology in the Universidade Nova de Lisboa, and her group is conducting translational research in the field of toxicology and regenerative medicine. One of the main research interests of her group is assessing the potential of stem cells for biomedical and biotechnological applications, and particularly to develop 3D organotypic in vitro systems for disease modeling and drug development. Joana is also a board member of the Portuguese Society of Stem Cells and Cell Therapy, is the national coordinator of the European Registered Toxicologists since 2017, and she's the chair of the Communication Subcommittee of Eurotox, and has served as coordinator of the toxicology section of the Portuguese Society of Pharmacology. Just a brief comment. If you have any questions, you can make them on the chat. And Joana, the title of her talk is Studying Liver Disease in 3D Liver Models. And Joana, the floor is yours. Thank you for being here today with us.
So, first of all, I would like to thank Javier for inviting me to be here today and to present some of the work that we have been doing in our lab over the last 15 years, maybe, already. The title of my talk will be Studying Liver Disease in 3D Liver Models, but I kind of try to give you an overview of the models that we have in the lab and show you some of the applications. So I will contextualize a little bit the presentation and then go deeper on the models that we have. The 3D systems that we have developed and the applications we already can do with these models, and finalized with kind of a finishing with all the challenges that we still face with these models. So the application of the 3D liver models is not only disease models, but of course it has a lot of application in drug development. And what we actually know that is that the novel drug discovery and development process brings a lot of challenges to the pharmaceutical industry, because they face a political pressure to develop safer medicines. At the same time, they should reduce the costs of this process. And despite all of the efforts for developing a safe drug that can take 10 to 15 years, the costs can go up to 1 billion euros. Still, at the end of the process, the success rate is quite low. And from the drugs that actually reach the market, around 18 to 30% suffer drug withdrawal due to hepatotoxicity effects. From those that reach the market, around half of those drugs did not show any type of concern in the preclinical or in the development process, in particular in the preclinical models, namely the in vivo models. And for this reason, a lot of effort has been done in the last years to develop relevant and physiological models that could help in the prediction of these adverse effects. The liver architecture, this is not new for you, for sure. The liver is a quite complex organ. It's composed of several cell types. So we have the non-parenchymal cells, that includes the stellate cells, Kupffer cells, cholangiocytes, and others. But the majority of the liver is composed by the hepatocytes that are the parenchymal cells. And those hepatocytes, although they morphological look all alike, depending on their location in the liver, they have actually different functions. So if they are closer to the portal triad, so they have higher access to oxygen levels in comparison to the ones that are closer to the central vein, and this is called the so-called zonation of the liver that gives the hepatocytes different functions. And of course, this will impact in their metabolism in terms of glucose homeostasis. So closer to the portal triad, they focus more in glycogen synthesis in opposite to the from lactate, whereas in the closer to the central vein, so the perivenous hepatocytes do the glucose homeostasis mostly from glucose. The lipid metabolism is also changing, the same for the amino acids metabolisms, and of course for the protein synthesis and xenobiotics metabolism. Therefore, when we think about developing a model, we also have to consider this, of course, that anatomically the liver is very well located, and for this reason this is also the major metabolism, it's the organ mostly responsible for the metabolism of xenobiotics and drugs, of course, and the idea is to detoxify the drugs or the xenobiotics that we are in contact with. The problem is that in many cases the compounds are not detoxified. On the other hand, they are bioactivated, giving rise to reactive metabolites that can be responsible for the adverse reactions that we see later on in the different individuals, of course. I just show this because this metabolic process, or the biotransformation process, is composed by several enzymes and systems, so enzymes or proteins that are categorized under phase 1, phase 2, and phase 3 classes, and this is just because later on when I show you the results, just to know that what do I call the phase 1, 2, and so forth. So the hepatotoxic mechanisms, of course, they are several. So we can go from mitochondrial dysfunction to oxidative stress, covalent binding of these metabolites with the proteins or DNA that will, of course, alter their action and function or damage the DNA itself. They can also promote changes or dysfunction in the tissue repair and in the immunological response. And all of these can be under the hepatotoxicity mechanism umbrella. So it's a quite complex process. And we also know that a lot of drugs are associated with some of those effects. So this drug-induced hepatotoxicity or drug-induced liver injury, DILI, can be an acute or a chronic response. It can also be classified as hepatocellular, cholestatic, or mixed, depending on their clinical presentation. Most importantly, it can be classified as intrinsic. It means that it's dose dependent, so it's predictable. This is something that it's an effect that we can predict, which is very well known in the case of paracetamol. But it can also be idiosyncratic. In this case, it's not dose dependent, and of course it is very unpredictable. And this is the reason why this DILI is largely unrecognized and underreported, so such that the true incidence is unknown. And for this reason, the concern and the need for liver models that actually are relevant and then can somehow help with the prediction of those effects has been so important. And for the people in the field, this was huge the day that the FDA for the first time said that we no longer need animal testing for the development of new drugs, as long as the data provided by the in vitro model is good enough, of course, and provides confidence enough to move forward in the process. So this is actually for the first time that it was assumed that the in vitro liver models can really have a role and maybe can make the difference, or that the animal models do not need to be performed if they will not provide further information. This is so important that the new was so important that it was even advertised in Science. So how can we actually create and better quality in vitro tissue models that can actually be microphysiologically relevant for this process? Several factors are driving this process. Of course, the first one is the interspecies differences that we always observe between animals and humans, the ethical concerns of performing in vivo studies, the need for cells in number and quality enough for doing or for establishing an in vitro model, overcome interdonor differences, and of course with the aim of trying to mimic the complexity of the systems, of the in vivo system in vitro, and to be able to maintain the cell's function in vitro. For that, different key aspects have to be considered when establishing a model. If we want to develop an efficient and fit for purpose in vitro hepatic model, we should mimic actually, or try to mimic or to consider the in vivo physiology, in this case of the liver. And so one thing that for many years was not considered enough was actually the matrix of the tissue. So people were mostly concerned about the cells, but did not consider the environment, the cells, and the extracellular matrix, for instance, where they are in the environment. So this is important because this matrix will influence the cell's access to nutrients or oxygens, for instance. And therefore, when we want to establish a system, we have to consider what type of cell sources we want to use. We have to consider the tissue, the architecture of the tissue, that of course will influence the flow dynamics and the formation of the molecular gradients. So as for the hepatic sources, we have several. The traditional ones were the hepatoma cell lines and so forth. Most of them already of human origin, which is great, but most of them also of tumor origin. So they do not mimic the physiology of the liver. So alternatively, other types of cells could be used. So we have the primary hepatocytes, of course, but the viability of those cells in culture is not good enough. They de-differentiate in vitro, so they lose their phenotype. But then we have the stem cells, of course, as a very good alternative. For the stem cells, we have different sources as well. So we have the IPS, the induced pluripotent stem cells, the embryonic, the mesenchymal stem cells, and others. They all have the potential to be differentiated in any cell type, including liver cells. And provided that we have a good protocol for the differentiation process. And of course, with that, we can overcome all the interspecies difference, the interdonor difference, and the low availability of human cells. This is some of the work that we already did in the lab in order to obtain hepatic cells from stem cells. So what we did was to try to mimic the in vivo liver, the development of the liver in vivo in vitro. So with different sets of cytokines, growth factors, and epigenetic modifiers, we could develop a differentiation protocol that follows this three-step differentiation process of the liver. So induce, or the first step consists in the endoderm commitment and foregut induction, followed by the hepatoblast and liver bud formation, and finally the hepatocyte differentiation and maturation. Of course, this was the result of a lot of work, but in short, this is a protocol that lasts for 24 days, so it's 1 month of culture to have hepatocytes, and then we try to extend the culture in order to have cells and cultures in enough time to make all the type of assays that we would need. After this process, we characterized the cells in terms of phenotypic markers, functional markers, and we also did the transcriptomic analysis to make sure that the cells that we obtained were hepatocyte-like cells, so HLCs. And of course, we used the corresponding controls. So in the global transcriptomic analysis, what we could observe is that we already, the HLCs here in the third column could already have already show the profile closer to the primary hepatocytes, or the golden standard, the HG2 cell line, and already quite different from the undifferentiated cells. We also observed that around 70% of these differentially expressed genes are changed by less than twofold when we compare the HLCs with the primary hepatocytes. But what I think it's the best results, and visually it's very easy to see that with this PCA analysis, that the HLCs are much closer to the primary hepatocytes than the HG2s themselves, and very distant from the undifferentiated cells. So it already provided us a good confidence about the quality and the phenotype of these cells. Then, of course, we went deeper with some gene expression analysis to make sure that we could have some very specific hepatic markers. So we tested the CK19 and 18 as a biliary and hepatic marker respectively, and we see, of course, a decrease of CK19 and an increase of CK18 throughout the differentiation process. So it's here in green, the differentiation. During the differentiation process, the cells are losing the CK19 and the CK18 is increasing, and the same for all the other hepatic markers, such as these transcription factors HNF4 alpha and CBP1 alpha. The same for plasma proteins, genes associated with amino acid metabolism, and also for the increased expression of some cytochrome P450 enzymes, so phase 1 metabolic enzymes. In some cases, the expression levels was even higher than in primary hepatocytes or HG2. The same markers were confirmed their presence by immunofluorescent staining, and we could also confirm that the cells showed the ability to store glycogen by PAS staining, and they already had a binucleated morphology, as expected for the hepatocytes. As for their capacity to produce albumin and urea, again, we could confirm that the cells were able to produce both albumin and detoxify ammonia through the production of urea. As for their metabolic competence, so the presence of phase 1 enzymes or phase 2 enzymes, this is very important in the drug metabolism process, of course. And we could also show that not only the expression of those enzymes was increased, but also their activity was increased, even when we induced the activity of these enzymes. And this was observed in all the HLCs after the differentiation, so after day 21. We also observed the presence of activities of CYP3A4, which is one of the most important enzymes in drug metabolism, still phase 1, but also the activity of phase 2 enzymes, and in all those cases, with higher activity when compared with the standards, so the EpG2 and the primary hepatocytes. So with this protocol, we could confirm that we were able to develop a differentiation protocol for obtaining hepatocyte-like cells from stem cells, and that those cells could be maintained in culture functional for more than 10 days. This was all performed under 2D conditions, of course, so we didn't mimic the tissue architecture. So after we wanted to move forward and to see if we could actually improve the functionality of these cells if we modulate the environment where they are cultured in. So, of course, when we think about the tissue architecture, flow dynamics, and formation of molecular gradients, we have to consider other aspects. The extracellular matrix, of course, the composition, the stiffness, it will influence the oxygen gradient and the other gradients. And actually, the truth is, if we want to, the ideal in vitro model should be a model that could simulate the physiological healthy, of course, tissue-specific or the pathological tissue-specific microenvironment. It should mimic the cell-to-cell and cell-to extracellular matrix contacts, because it will, of course, influence the cell shape and polarity and all the environment that is surrounding the cells. So as for 3D cultures, a lot has been done over the last decades. Since the 60s, that was actually the first time that the 2D systems of animal cells were reported. Much has been done, going from the traditional 2D and passing by the 3D systems with spheroids in scaffolds. Then it could be the complexity was increased using co-cultures. Then using the dynamic systems, using bioreactor systems, moving to multi-tissue cultures, and finally, nowadays, we have the so-called organ on a chip, where we can have the cells communicating in a miniaturized environment. All these systems are available nowadays. They all have several advantages, but the idea is always to recapitulate the complexity of the microenvironment of the cells in vivo and to reduce the gap between the cell cultures and the physiological tissues. In the liver, much has been done since the sandwich cultures, which actually was quite important by then, because for the first time we could polarize the hepatocytes as we have in the systems. But they are not, nowadays, most of the work with the liver cultures is actually on using the spheroid systems, the organoid systems in static or dynamic conditions, and of course using the bioprinting and the microfluidic platform technologies. All of them, there is not the perfect model, I would say. All of them have advantages and disadvantages, but of course, the more complex is the system, the higher costs are associated with, and I think that the most difficult thing actually is to have personnel trained to work with this complex system, because it's not that easy to perform. In the lab, we have been working with many of those systems actually. We work with spheroids and organoids in static or dynamic conditions, and also we already started working the microfluidic platforms. So we started with quite simple systems using primary red hepatocytes, moving forward with a little bit more complexity using the bioreactors. We can actually fully control the cultures. And have a controlled environment. Later on, using the static spheroid cultures, we use the stem cell derived cells, and now we move to the microfluidic platform. I will give you some examples of these works, not too much in detail, but just to show you the relevance of the models. And one of the work is this that we performed some years ago with nevirapine. We used the nevirapine as a model drug. So for you, of course, you know that this is an antiretroviral drug. It's not very much used in Western countries, but in developing countries still very much used due to its efficacy. But we chose this drug not for this, but because it was approved in '96 in the States, and in 2000 there was a boxed warning issued by FDA due to severe life-threatening and fatal cases of hepatotoxicity. It is interesting to test in a model or to validate the model because nevirapine is bioactivated. So upon its metabolism, there is the formation of reactive metabolites, and it's a consequence of the nevirapine metabolism that has a quite complex not that complex, but it's metabolized by phase 1 and phase 2 enzymes. In phase 1, we have several hydroxy metabolites, the 2, the 3, the 8, the 12 hydroxy nevirapine, and the carboxy nevirapine as well. Those are further metabolized, and there is the formation of these sulfoxy or these quinone methides that are actually responsible for the toxicity associated with nevirapine. So if we could see all of this in the in vitro model, it somehow would validate the model, and that's what we did. So we set up these 3D dynamic cultures of hepatocytes, and with the 3 days difference, we added to the cultures nevirapine, or one of its metabolites, the 2-hydroxynevirapine and 12-hydroxynevirapine that we know are associated with toxicity effects. So, and what we could, it's important to tell you that also not only nevirapine, but also its metabolites, they are auto-inducers of their metabolism. So it means that they induce the activity of these phase 1 and phase 2 enzymes. And we could show here that the changes in the activity of those enzymes, and we can see it via ECOD, EROD, and SULT1A1 activity. The activity of those enzymes was increased upon incubation with nevirapine or any of those 2 metabolites. This could be observed in the 3D system. We could also observe only in the 3D system versus the 2D cultures that the same profile that we see in humans is also observed in the 3D system. So we have the formation of all nevirapine, phase 1 nevirapine metabolites, and in the same proportion, so higher amounts of the 12-hydroxynevirapine followed by the 2, 3, and 8, and also the formation of the phase 2 metabolites, the ones responsible for the hepatotoxic effect of nevirapine. And this could only be observed in the 3D systems. Later on, we could also observe the formation of protein adducts of nevirapine, and those adducts could also only be observed in the 3D system and not detected at all in the 2D cultures. And with those results, we could validate our model and show the relevance of the 3D systems when compared to the 2D cultures for drug metabolism studies, and of course they can also be used for biomarker identification and so forth. We performed other works in parallel with these ones for validating the system. One of those was to evaluate PON activity, peroxidase activity, so it's a marker of hepatotoxicity. And also we could only observe a change in the activity of this enzyme in the 3D system upon incubation with nevirapine. And another interesting work was this one that we performed with sample patients. So we compare the data from the 3D and 2D system with data from patients and the changes in ApoA1 activity, for instance, that is observed in patients that are under treatment with nevirapine could also be observed in the 3D system and not in the 2D. So further validating the relevance of the model. All of this work was performed with red primary hepatocytes by then. So we wanted to move forward and use human hepatocytes. For that, of course, we had to use the HLCs that we already developed the protocol. And so we tried to adapt these cells to different 3D systems. So to static conditions, to a perfused hollow fiber bioreactor, and compared with the 2D monolayer culture, of course. The objective was to see if the modulation of the culture conditions would actually influence the phenotype of the cells. As expected in all the systems, we could detect the specific hepatic markers, the ones that I already talked about earlier, albumin, HNF4, and CK18. Phase 1 and protein transporters were also detected in all the 3D systems. And the capacity to store glycogen was also observed in all the systems. Then when we go deeper to see other type of functionalities, such as the production of urea or albumin, then we start to have different results. So the system that showed higher capacity to produce urea or to detoxify ammonia was the static model, so the spheroids. And as for the albumin production, the best condition was the bioreactor system. As for the metabolic competence of the cells, namely the capacity to convert bupropion or diclofenac, again, I'm sorry. Again, we could already observe some differences between the spheroids and the bioreactor, but when we test, for instance, the IC50 in the 2D versus the 3D systems, we can have diclofenac in particular. We see low EC50 in the 3D systems, meaning that the cultures are more sensitive to diclofenac, which makes sense because the toxicity from diclofenac comes from phase 2 metabolites, and so the cultures have to be competent to produce the phase 2 metabolites as it happens in the 3D system. So after this, what the final part of the talk is to show you some of what we have been doing with the microfluidic devices. So the microfluidic devices are a system, a miniaturized system that has the advantage that we can design fit for purpose. We can do whatever we like. It enables the communication of different cell types. We can include dynamic flows, and of course it consumes very low amounts of cells and samples. The disadvantage is that when working with so few amounts of cells and samples, it's also very problematic in terms of analysis of the cultures. And of course, everything changes when setting up the cultures. But the truth is, it helps in recapitulating both the in vivo hepatic function and architecture. We can do it in the microfluidic devices, and some work already proved that it can help with the culture of hepatocytes. So we tried to, the idea was to adapt our HLCs to the microfluidic devices and see how we could use these cells later on for disease modeling, because, oh, I'm sorry. I don't know, but whatever. The microfluidic devices are a very good system if we want to induce a disease in the system and to understand what how the cells communicate in this condition. So that's what we actually wanted to do. But for that, and before putting the cells in the microfluidic devices, we had to make some adaptations, because the truth is most of the culture media that is used to maintain the hepatocyte cells in cultures are very rich in glucose, insulin, and dexamethasone. Nothing of that is physiologic. So if we want to induce insulin resistant or even muzzled, we should bring the cells to a physiological state before inducing the disease. So the first step of our work was to adapt the cells to a more physiological condition. And then later on, adapt the cells to the microfluidic device and try to develop disease model of muzzle. So the differentiation medium contains high concentrations of glucose and insulin. So what we tried to do was to reduce these concentrations to more physiological levels and to reduce the concentration of dexamethasone. Not for a physiological level, of course, but at least to decrease the concentration of dexamethasone. And then we compare the two conditions, so the differentiated medium versus the physiological medium. So we call it physiol versus diff. And we didn't see big changes in mitochondrial function via the seahorse analysis. We could see that the cells maintain their ability to store glycogen and to produce albumin and urea. And again, they still maintain the phase 1 and phase 2 enzymatic activity. When we did also an IC50 for paracetamol, both in physiological and differentiation conditions, in diff conditions, we could also see a decrease in the IC50 of paracetamol, which already give us a hint that maybe the physiological conditions are actually better for maintaining the cells. Later on, we then adapted these cells under these conditions to the microfluidic devices. This is a whole new world. We have to learn how to work with cells again, I think, when doing the microfluidic devices. It's totally different from the other systems. We have to learn, but we can do it. So we had to optimize everything again, the coating, the inoculation conditions, and so forth. Once it was done, we then tested the cells if they were responsive to insulin and glucagon. And so what we could see is that under these physiological conditions in microfluidic devices versus the 2D traditional conditions, we already can see that the glycolysis is induced, bile acid metabolism is induced as well, and that the neogenesis, the fatty acid metabolism, and the mitochondrial function is inhibited. As for the glucagon response, we see exactly the other way around. So glycolysis is inhibited, bile acid metabolism is inhibited, glycogenesis is induced, glyconeogenesis is induced, fatty acid metabolism as well, and mitochondrial function is induced. So it somehow give us some confidence that we would have cells responsive to these different factors. So we tried later on to see if we could somehow induce a muzzled disease model in this system. So we use different concentration of fatty acids, and we could see that a dose-dependent response, so higher concentration of fatty acids, higher incidence of lipid droplets. And we also see a change in the glucose metabolism with an induction of gluconeogenesis along with higher glucose production. We also see changes in the cell's metabolism, so we see an increase in the lipogenesis and a decrease in the spare respiration and increase of proton leak linked respiration, so it's showing some mitochondrial dysfunction under fatty acid incubation of the cells. And also an increase in ceramide synthesis and of inflammation markers, showing that somehow we are already having some markers of associated with MASLD. So I would not say that we have a MASLD model in vitro, but at least we already have some markers showing that we are inducing some stress and inflammation process in the cells. So with all of this, I hope that I could have showed you that the stem cell derived cells have a very good potential to study for providing in vitro systems for toxicological studies or disease modeling. That actually this 3D system is a huge field with variety of applications. That it's actually a challenge to create these systems, although they can be relevant and can recapitulate the microenvironment of the cells. And again, that there is not a perfect system, that we should choose our system according to the purpose of our study, and that they can actually help the regulators and in taking decisions later on. So what I think it's where we stand is that actually these models do have a role in the prediction of clinical conditions. We can integrate the data from non-clinical assays and help and use this data for extrapolating the results. And that is it. All of this work was performed not by me, of course, but all the people in the lab and via the funding that we have been having over the last years. Thank you. We have different controls that I think we should also always consider in these models. Either a culture control, meaning if you are using a 3D or whatever, you should always use a traditional system as a control to confirm that your system is providing better results or not. I mean, to show the differences in the systems and the cells. So this is the other control, because now, I mean, I only showed the results with hepatocytes, of course. So if we consider hepatocytes as an example, taking this example now, of course, if you derive the hepatocytes from the stem cells, you should use undifferentiated cells as a control as well, because the stem cells, they themselves have a metabolism, and of course you have to understand if the results are already coming from the differentiation process or not. And the primary cells, so the ones that are isolated directly from the liver, because actually those are the golden standard. That's where we want to reach. So if you isolate the samples from the patients, that should be performed. I mean, the human cells are always isolated from the patients. The thing is that we can have access to those cells commercially. If they are commercially available, usually they are
disclosed.
Exactly. Otherwise, you have to do it yourself. This topic has been very hot some years ago. So people were working hard, and this bioreactor system, this one that I show here, actually was developed initially for having bioartificial liver systems. it was a very good model because we can mimic the blood flow. These hollow fibers were specifically designed for that. This was a collaboration we had. So they were the ones developing this bioreactor. The thing in the end is, one, is almost impossible to have the huge amount of cells we need to provide, to have a bioartificial liver, okay? The only way we managed to have the cells in quantity enough was cells from porcines, not from human, because it's too much. Even from deriving the cells from stem cells, it's millions and millions of cells that we need, so it's too much. So, but again, it doesn't have to be a problem to use porcine cells, okay? But from the practical point of view, so to really apply these bioartificial systems and models into the clinical setting, in the end, I think it's not feasible, actually. So I don't think it will work. That's why it's not such a hot topic nowadays, because I think people realize that actually it's not easy to develop bioartificial livers that we can actually use in the end. Never worked. But yeah, for sure. But, okay, so liver regeneration is a different thing, and when we want to induce liver regeneration, then we have to consider the different cell types. And I think when we want a model for liver regeneration, should not be only hepatocytes. But I mean, we have to start somewhere, so we can start with hepatocytes and then maybe increase the complexity of the model, and of course include the Kupffer cells. They play a role, definitely. Maybe those two, the first two more important cell types, I would say it's the hepatocytes and the Kupffer cells, but then you have to have the stellates and the others. But yes, the organoids would be great for that, I think. I don't think you should compromise complexity if they provide you the results you need. I also don't think that you just should make the system complex just because, if it will not enable you to get the answers you need. So people think that because I work with 3D models, I always think that 3D models are the only ones that should be performed. That's not true, okay? In many cases, the 2D are more than enough. So, okay? So the thing is, when you do when you increase the complexity to include the different cell types, for instance, you have to consider that if the cells are in an organoid, for instance, so they are co-cultured together in direct contact, the effect of the different cells may not be distinguished. They are all there. They have the effect that is produced in the cells is there. So you have the final results, but you may not understand which one is triggering what. So this might be a problem, depending on the information you want to obtain. But that can be overcome using a microfluidic devices with the different cells in the different systems, or with the co-culturing in transwell systems that can also work very well as well. And we did some work in the transwell systems already with Kupffer cells and hepatocytes to see the paracrine effect of the cells, if we can modulate and how do they respond to each other. So if we trigger the Kupffers, if we have an effect in the hepatocytes. So only this type of cultures can provide us this type of results. Otherwise, you have to have the medium conditioned by one cell, take this medium and put this medium in contact with the other cell type. It can also be done. But then there is no communication. It's effect, but separate effect. If you want to communication, then you have to have this system. So for that, it's I think it's the microfluidic devices that can help that, because we can design the system as we prefer. So we can increase the blood flow, or the fluid flow, not the blood flow, or we can decrease the pressure of the flow, or we can increase or decrease the size of the channel where the cells can be cultured. So I think it's actually with the microfluidic devices. At the hepatocyte level, so it's you have to go from to different genes, maybe, that are involved in these pathways, because there are so many things that are changed in these conditions. So all these functions can be altered, even the capacity to produce albumin or urea can be changed. So the fatty acid metabolism, the lipogen, I mean, there is a pack of things that has to be actually tested. There is not one endpoint. No, I didn't look. For that, we would have to have endothelial cells as well in the system. It has been done, but we didn't. Definitely, yes. We should have harmonized techniques and systems to endpoints to characterize the cultures. I mean, even for something so simple as the phase 1 and phase 2 activity. If you go to most of the papers, they only measure gene expression, doesn't measure activity, enzymatic activity, you know. So actually, the more you characterize, the better. But it would be better that if we have at least a set of endpoints that we should have, the minimum endpoints to show that the model is actually relevant, or at least works for your purpose.
Thank you, Joana, for this excellent talk. Thank you for being here today. We will have a brief recess of 5 minutes and we will be back soon.